much enjoyed some of the previous presentations at this meeting, and I'm grateful to have the chance to share some of the work that we're doing here at Sick Kids in Toronto at the Mouse Imaging Center. Both John and I work quite closely together. So I'm going to start by telling a tale about some of our work looking at how chemotherapy treatment impacts brain development. And I'll start by talking about some of the motivation from the clinical side, looking at brain function and structure and survivors of acute lymphoblastic leukemia, and then go into our work in a mouse model to look at how we look at the treatment effects and some of our initial work at trying to understand the mechanisms that underlie those effects, of course, with the long-term aim of taking that full circle and trying to improve outcomes. So the first small study I'd like to talk about is one we did in a population of survivors of leukemia. So acute lymphoblastic leukemia is the most common childhood cancer and represents something like 30-ish% of childhood cancers. It's mercifully not extremely common. So at Sick Kids, we see something like 60 patients per year. So we did a study looking at 71 ALL survivors about 8 to 10 years off treatment. They were all treated with only chemotherapy, and we wanted to look at their outcomes in terms of IQ, academics, motor, behavior, and perhaps more importantly, in terms of a qualitative perspective, their quality of life. And we also did MRI in these populations. And so just to give a quick view of some of those IQ measures. So we ran the WISC, which is kind of a typical intelligence quotient type of measure for children. And it gives you 4 different readouts. The ones I'll focus on are either the last 2 here, which is the working memory index and the processing speed index, where ALL survivors are shown with blue points and a control group of typically developing individuals is shown in orange points. So on this scale, you'd normally expect your population to be about 100 with a standard deviation of something like 15. And in the ALL survivors, we see, they're both smaller than or performing more poorly than the control group, as well as lower than would be expected from population norms. And we looked at, I'm not going to go through all of the different measures we did. There were mild motor impairments, as well as poor performance on at least math-related academic outcomes. We also, though, asked more generic quality of life. And for this, we looked at what's called the Pediatric Quality of Life Inventory, which is a very simple instrument that's given both to parents and to kids. I'm going to talk about the proxy version, which was given to parents. And it has 23 very simple questions. One category is on function at school, and it has simple questions like, how do you have trouble paying attention in class? Do you have trouble with forgetting things, keeping up with schoolwork, and so forth? And a very simple Likert scale where you select never to almost always. So it's not, I like to emphasize, it's not, how well do you perform in advanced calculus? These are sort of very simple measures of school function. And if you look at that across the 2 populations, where I'm showing the control group on the left and the leukemia survivor group on the right, you can see this is just a histogram showing the proportion of individuals in each of these kind of score bins, where typical scores would be quite high for quality of life. So 100 would be no impairments of any kind, and very low score would indicated almost always have problems with all of these measures. So you can see a definite shift in the survivor cohort, where they're definitely, the majority of them are showing up in the second of these bins as opposed to the first, and you've definitely got a tail running through. So overall, quality of life. And this was the most dramatic of the different quality of life scales that the PIDS-QL produces, but also an important one, and all of the metrics showed poor outcome. So we also looked at MRI in this cohort, the same cohort of 71 survivors, and compared that to the same 80 or so controls. And I'm just going to talk about brain structure today. And we saw pretty widespread volume decreases in the ALL survivor group relative to the control group. So all of the colored regions here were statistically significant in being smaller, and some regions, particularly some of the white matter, showing a higher volume loss relative to the controls than gray matter, but all the regions really showed significance, with the exception of the occipital lobe, which seems to have been pretty much spared compared to the other brain regions. So this is typical, I think, of not just our group, but what other groups have seen, where ALL survivors have a higher risk for poor IQ and academic problems. And that's usually where things start to appear, is more difficulties in school, and then parents and get worried about how children are doing. Also have a higher risk for behavioral problems, particularly inattention and learning related problems. And this broadly impaired quality of life, accompanied by brain structure. So one of the things we really want to do is, of course, ask, well, why are these impairments occurring, and what can we do about them to optimize outcomes for the leukemia survivors that were able to cure of their cancer? It's actually a very difficult question to answer. The chemotherapy protocol is quite complicated, and so it's really easier to try and look at it in a mouse model, and I'll do that in the next slide, just sort of relating it to the human protocol, which is quite complex. So here there's kind of two rows on this graphic that I'm showing here, where the top row is what we would call a standard risk ALL protocol, and I'll focus really on that. But you're not meant to take much out of this, other than it's quite complicated. The phases of treatment all include multiple chemotherapy agents. They last for quite some time, and the whole treatment runs for a couple of years, which for children that are diagnosed at 2-3 years of age, by the time they're done treatment, they've been on chemotherapy for a majority of their life before they achieve a cure. And all of this protocol has really been the development of it has been motivated by survival. So you want to make sure that children survive. And so it's very difficult retrospectively to look at this and say, well, what part of this treatment is causing problems? And in approaching it, it gets right into a common problem, I think, in assessing animal models of this. Obviously, you're not going to change the treatment in children, so we want to model this treatment experimentally in mice. And it becomes quite a big problem in that, in any mouse experiment, you're going to have some number of mice per group. You're definitely going to want to look at this in both males and females. But there's also questions of maybe a genotype might be of interest. And we, as I've just highlighted, there's many different types of chemotherapy agents, and you probably want to look at each of them, if not their combinations. Behavior of those treatments might be different in one strain versus another. So, you can imagine that as you start to slot the different conditions you'd like to probe with each of these, it's all multiplicative. So even in very simple experiments, you can very quickly get up to a scenario where you need hundreds of images. So we've been addressing that at the Mouse Imaging Center by a strategy we call multiple mouse MRI, where really we just take multiple RF coils and house them in the magnet together, and with some effort on pulse sequence modifications and data wrangling. You can set up your imaging strategy, so you can set it up and scan as though there was just a single mouse in there. But the idea is that at the end of that scan protocol, you'd have n images produced based on how many different mice you're able to put in the magnet. So we have a couple different systems that are intended to be able to accommodate that type of scanning. So here's our 7030 magnet, which was installed in 2016. It is equipped with 4 separate cryoprobes, and you can see they're just in a 2x2 array here. And so there's 4 mouse sleds on the table in front of the bore here, and you can mount a mouse in each of those and slide them all in simultaneously. And the idea is then you'll get 4 images at the end of your scan time. And that's hopefully for the price of one, so to speak. We also more recently have, and this is an example of some of the images we're able to get. So this is a little bit higher resolution than we usually run. So here's a 60 micron image that's an average of 25 or 30 different mouse brains. And you can see quite exquisite detail. This one's been acquired with a T1 weighted sequence and manganese enhancement. And you get quite nice delineation of the cerebellar nuclei. You actually can see some of the layers in the colliculi and other features that are sometimes difficult to see in lower resolution versions of these scans. We also have a second system that was about a year old now, and this is an 11722 system, and this is showing what it looks like as installed in place of the former variant system that we had here. And it's got a smaller bore, but equipped with more channels. So all the data I'm going to present today are from the former system, but we're certainly hoping that this system will be the workhorse for many of our projects going forward. So going back to our ALL treatment, given the complexity of it, one of the first questions we want to ask is, well, how do each of those chemotherapy agents affect long-term outcomes in our mouse model? And so here I'm just listing 8 representative chemotherapy agents from that broader pool of chemotherapy agents used in ALL treatment. And the idea was we wanted to take each of these 8 agents and treat our mice and ask how did this affect their outcomes in terms of brain structure. So here is the rough timeline. So mice were allowed to age to postnatal day 17. Then were given their first systemic chemotherapy treatment. And then a couple of days later, they were given a second dose. This is a relatively juvenile age of treatment, but of course, we're trying to model pediatric treatment in kids. And so that was a deliberate choice. And then we monitored them up to an early adulthood stage, P63, and then in this case, looked at them with ex vivo MRI. So sort of one slide summary of a lot of work that went into this is shown here. So there's 4 columns on top and another 4 on the bottom that represents the 8 different chemotherapy agents that we treated with. And I've sorted them here sort of at the top left from those that show the most similar volume decreases at this early adult stage to what we were seeing in the ALL survivors, to the lower right, which is sort of least comparable to the phenotypes that we saw in the ALL survivors. And so we see these top 3 here, vincristine, doxorubicin, and methotrexate, which show pretty similar volume losses and comparable to the ALL survivors. So these were the ones that we prioritized as being most relatable to the human outcomes and of interest. Methotrexate arguably would be the one that I think most hematologist oncologists would have said that's likely to be the problem. But this also flagged doxorubicin and vincristine as potential candidates. And I'll talk about those in a little bit more detail in a second. So this gets us to the next stage. We said, given these sort of 3 different agents that we think are important, what might be the mechanisms that these individual chemotherapy agents work by? And it's interesting in that it's thought that some of the toxicity mechanisms may be different than some of the anti-cancer mechanisms, which would be a good outcome because that would give you a window to potentially change treatment or intervene to try and improve outcomes without, obviously, the higher priority is making sure that anti-cancer efficacy is still present. So understanding, what the mechanisms are in each case and how they may differ is quite important in terms of being able to translate some of these findings. So I want to talk about each of those three agents in turn, and it'll be kind of a whirlwind on each of them, but I think the examples of each are informative. So the first one is methotrexate, and as I mentioned, this is one of the more important chemotherapy drugs. I've highlighted on the standard risk ALL table that I showed earlier. In red, the methotrexate treatment that's delivered intrathecally. This is an injection into the CSF space around the spinal cord, and it represents the CNS targeted part of the ALL treatment. But it's also delivered systemically, and that's highlighted with the blue circles. So that's done in a few different stages, and mostly in this high dose IV format, but also at lower doses in an oral format for an extended period through the last couple of years of treatment in what's called the maintenance phase of ALL treatment. So methotrexate it functions by inhibition of folate metabolism, and it was actually the first chemotherapy agent really showing any success with ALL, and it first was reported in the late 1940s and very quickly became the backbone of leukemia chemotherapy, and is commonly thought to be associated with potential cognitive impairments. So one of the first things we wanted to ask is, well, obviously it's given by two different routes. Does the systemic route versus the intrathecal route make any difference in terms of how the brain is affected? So the first challenge to that was really asking, how do we do this intrathecal injection in a young mouse? The mice are quite small, and the target region is even smaller. So intrathecal injections are difficult in mice, even at adult stages. So what we ended up doing for that is graduate student Sunny Choi developed a protocol based on ultrasound guidance of the intrathecal injection. So I'm showing here on the left side ultrasound images where the mouse is on its side with the back pointing to the left of the screen, and the ultrasound probe is at the top of the screen. And she was able to design this protocol where basically at the bottom end you can see the pelvis of the mouse and the ilium, and walking up you can find the fifth lumbar vertebrae, and partway between those two you can locate the target region where the needle can sneak between those vertebrae and catch the intrathecal space. And as sort of proof of concept, she did some ex vivo MRI beside that just to help interpret our ultrasound images. But through that protocol, she was able to get quite a high success rate in fairly young mice in delivering the chemotherapy. And so through this method, then we did a study where we treated mice both systemically and intrathecally in separate groups, and asked how do these two different groups compare in terms of outcomes. In a one-side format here. So interestingly, I'll start by looking at the two color-coded images at the top here. So this is just showing a average image in the background showing the mouse brain, and then overlaid on top of that in colored regions are those regions showing a statistically significant difference between the groups, where blue is showing this volume loss, which is what we would expect. We saw a much wider spread volume loss after the systemic route. And we've also noticed that the IT route really has these more focal regions and needed a remarkably high dose to achieve these volume changes. So the volume changes shown here were achieved only when you had a sort of dose into the intrathecal space that was getting close to comparable in terms of mg per kg to the dose that was delivered intravenously in the whole animal. So it seems like the IV route was producing a much broader change. So summarize that on the bottom here is a plot just showing total gray matter volume, total white matter volume. And you can see the IV route is producing a larger sort of impact on the brain than is the intrathecal route. Interestingly, to kind of understand the distribution of the volume changes in that intrathecal route, Sunny did an injection of gadolinium contrast agent into the intrathecal space, and then from the ultrasound scanner ran over to the MRI scanner and asked what does the distribution of this gadolinium injection look like, and that was sort of just evaluating effective delivery of drug to the brain. And you can see the distribution of the gadolinium in this T1 weighted images, where you see the ventricles, as you would expect, particularly the fourth ventricle, which it seems to get the gadolinium more quickly, as well as then the front of the brain, close to the olfactory bulbs, which also lights up fairly quickly with the gadolinium distribution. And that pattern really is replicated in the volume changes that we're seeing, where the biggest volume changes are occurring around the fourth ventricle and up in the frontal lobe near where the olfactory bulbs are. So it seems to be consistent with the distribution of the CSF from the intrathecal space after an intrathecal injection. So that is interesting in its own right, I think, because it really refocuses our attention that methotrexate, even though it's targeted to the CNS through these intrathecal injections, may be having a bigger impact through its systemic delivery route than through a CNS targeted route. The next agent we're interested in is doxorubicin. So doxorubicin is an anthracycline. It's through all of the phases of ALL treatment, although it is a much more common agent in breast cancer treatment and a very important agent there, and in some instances has been associated with brain fog and problems in adult cancers. So it's still a candidate that we're interested in, both because of our preliminary data as well as that sort of corresponding data in penetrate the barrier particularly. So our hypothesis there was that it was probably having an impact systemically, and that systemic impact was then in turn having an effect in the brain, which based on our methotrexate results may be true for methotrexate as well. But we started at that point with doxorubicin, and so one of the first studies we did was we characterized a panel of different cytokines and asked what cytokine changes are occurring after administration of doxorubicin, and this was measured in the blood. And there's a number of changes. One of the more prominent ones, at least early after early after treatment, was IL-6, where within a day of treatment, you saw quite large changes in IL-6 in the doxorubicin group, quite variable, but many-fold. And so this is a large change. So we asked the question, is that IL-6 level having any impact on the outcomes after doxorubicin treatment? So we took an IL-6 knockout mouse and looked for brain phenotype changes after doxorubicin treatment in that IL-6 knockout mouse compared to litter mate controls that were wild type. So on the top here, in this figure, I'm showing doxorubicin-induced volume loss across the brain. This is fairly widespread, as we reported earlier. This is at an earlier time point than P63, but otherwise similar in terms of the display. So quite widespread changes in volume. And in the IL-6 knockout mice, which is shown in the second row, I'm showing a display here of percent rescue, where 100% rescue would mean 0% volume loss after the rescue, and 50% would mean you sort of spared half of that volume loss. And you can see several regions, not everywhere in the brain that's affected, but several regions in the brain where you're actually getting reduced volume loss, and in particular regions that can be quite prominent. So in the fornix, white matter structure, you're seeing near complete mitigation of that loss in the knockouts. And likewise, the primary motor cortex is, almost normal in the knockouts compared to the wild types where there was a definite volume decrease. So IL-6 does have a role. It doesn't seem to explain all of the volume losses across the whole brain, but certainly seems to have an impact that's important in determining volume loss in several regions of the brain. So the last agent I want to mention here is vincristine. Vincristine, like methotrexate, is quite an important backbone in ALL treatment. It is given at every stage of treatment, starting right at the beginning and running right through the end. And it works by disruption of microtubules. If I go talk to my hematologist, oncologist colleagues across the road, they will tell me that vincristine is often associated with peripheral neuropathies, so sort of tingling and sensation changes in the fingers and toes, and sometimes in more prominent cases, actual, fairly prominent pain, but is less commonly associated with any changes in the brain. So this is really one where our initial study looking at different chemotherapy agents highlighted this as a potential candidate that I don't think would have been identified necessarily clinically. So there's still reason, though, to think targeting this would be interesting. So in the context of peripheral neuropathy, the thinking is that this microtubule disruption is damaging to axons. And there's an important action of SARM1, which is involved in sort of axonal degeneration after initial injury. And this axonal degeneration, certainly peripherally, is a concern for some of the peripheral neuropathy phenotypes. And it's been reported as presented here relatively recently that knockout of SARM1 can actually reduce or eliminate the peripheral neuropathy as seen in mice. So we wanted to ask, does this same axon pathology have any role in the vincristine-induced changes that we're seeing in our mice after vincristine treatment? So likewise, I'm going to show a very similar slide layout to the one I showed for doxorubicin here. So on the top, I'm showing the vincristine-induced volume loss. Again, this is a little bit earlier than the P65 time point, but the same format of the layout. So fairly widespread volume losses induced by vincristine treatment. But we, in SARM1 knockout mice, are able to eliminate a fairly broad portion of that. So much of the brain that was affected by vincristine is, at least in part, rescued in these SARM1 knockout mice. Not the rescue isn't full everywhere, that's for sure, but the plots on the bottom show the examples of the corpus callosum and the hippocampus, and both of those structures are largely spared in the SARM1 knockout mice, at least as compared to the wild type mice. So I think this is an example of some ways we can use chemotherapy treatment, use MRI to give insight into the action of chemotherapy treatment and its toxicity in the brain. So just to wrap up here, our work has shown in humans that AL chemotherapy does have some neurotoxicity, and the use of our animal models and MRI has been providing sort of an ability to dissect the treatment itself into its component parts, as well as then with those individual components try to gain insight into how that toxicity comes about. And of course will also give us insights as we sort of assay interventions as to how we can make those outcomes a little better. So I'm grateful for everybody's time. I'd like to thank my colleagues at Mice. In particular, I talked about the work of Sunny Choi, who did the methotrexate work with the intrathecal versus intravenous comparisons, and Jonas, who's actually going to talk next, who did the work with both doxorubicin and vincristine, as well as the other colleagues on this slide that have contributed. Thank you.